Hello everyone, welcome back to Dr. K. Primer lecture series presenting by Dr. K. Prem, that's me. Today we will talk about gram staining procedure and the developers of the gram staining. Here you can see two people, Dr. Hans Christian Gram and Carl Wiegert. Dr. Hans Christian Gram was a Danish pathologist. Is from Denmark. While he, in 1984, while he working with the lung tissues from patients who had died of pneumonia. So he collected a fluid sample from that lung tissues and smeared on the glass slide, and he applied certain stains and examined under the microscope. And for surprisingly, you could see that certain bacteria preferentially retained the color he applied. Other bacteria could not retain the color. The bacteria which could retain the color called as a gram positive. They looks purple under microscope. Then later on, Carl Wiegert introduced a counter stain, that's a saffron, to stain the bacteria which could not retain the primary color, and those bacteria could look pink under the microscope, or pink, red or pink. So they are called as a gram negative. So these are the two people who have invented the gram staining procedure. As you all know that gram staining is the first and foremost used method to distinguish the bacteria into gram positive and gram negative. And the gram positive and gram negative is all depends on the thickness of the cell wall. If the bacteria have thick cell wall, it can retain the primary color, right? Then that is a gram positive. Whereas a bacteria which had thin cell wall, which could not retain the primary color after discoloration or decolorization, then it can be colored with the counter stain, that's called as gram negative. Here you can see a few diagrams where I want to show the thickness of the peptidoglycan or cell wall between the gram positive and gram negative bacteria. You see, in gram negative have three layers. That's a outer membrane, which is mainly have a lipids, and the peptidoglycan, that's a cell wall, and a thin cell wall, then a plasma membrane. Then whereas gram positive do have thick cell wall, consists of 90% of the cell wall is a peptidoglycan, and then plasma membrane. So as I told you that retaining of the color is all depends on the cell wall thickness. The cell wall mainly consists of peptidoglycan. So here in the next slide, I want to explain the chemical composition of the peptidoglycan. As you all know that peptidoglycan consists of polysaccharides. Each polysaccharide have one disaccharide is polymerized. Each disaccharide consists of NAG and NAM, N-acetyl glucosamine, and N-acetyl muramic acid. N-acetyl muramic acid had pentapeptide at third carbon. The disaccharide of NAG and NAM are synthesized in the cytoplasm and exported to the external surface of the plasma membrane, where it is polymerized by using penicillin binding proteins. So polymerization of the polysaccharide, that's NAG and NAM, is catalyzed by the enzyme called as glycosyl transferase. So glycosyl transferase establishes the uh, glycosidic bond, that beta 1,4 glycosidic bond. Then the two strands of polysaccharides are cross-linked by the peptide bond. The peptide bond between the pentapeptides of one strand and pentapeptide of other strand is catalyzed by the DD transpeptidase. DD transpeptidase. DD transpeptidase forms the peptide bridge between the D-alanine of one strand and L-lysine of other strand. The cross-linking is very, very important for strengthening of the cell wall. So the glycosyl transferases and the transpeptidase both are belongs to the penicillin binding proteins. As I told you that the thickness of the cell wall will determine the whether that is gram positive or gram negative. So based on the thickness only, they have a ability to retain the primary color or loses the primary color, right? So gram staining had 5 steps. The first one is fixation, addition of crystal violet, and mordant, fixing the primary color with the mordant treatment, that's iodine treatment, and decolorization with the alcohol or acetones, and addition of counter stain. In this one, you have to be very careful what is the age of sample. If you take overnight culture of bacteria, usually you get the accurate information. If you take a refrigerated culture, gram positive can be looked as a gram negative may be looked as a gram positive. So that's why, so overnight culture is ideal sample for gram staining procedure. So whenever you take a sample of the bacteria, you can smear on the glass slide and fix with the flame. That's a fixation. Once you fix the sample, right, add the crystal violet color. Crystal violet had crystal violet and chloride molecules. When you prepare a crystal violet aqueous solution, crystal violet and as a crystal violet and chloride ions, they'll be dissociated. Crystal violet is a positive, positive chloride is a negative. Crystal violet can bind to the negatively charged molecules in the bacteria and bacterial cell wall, and that the color will be retained. Then to fix the crystal violet color, you can add iodine, which acts as a mordant. So iodine can form a associated with the crystal violet, form a large complex called as a crystal violet iodine complex. That complex cannot come out of the cell wall, and it will be there with the only cell wall and the bacterial cell. Once you are done with the iodine treatment or mordant treatment, then you can go for decolorization with the 70% of alcohol, or otherwise you can go for acetone, where the color will be removed with this treatment. If color is not removed, then the color is retained. Then the bacteria, if they look purple color, they are called as gram positive bacteria. Other sample where you do the same treatment, like fixation, crystal violet addition, iodine treatment, and decolorization. When you do the decolorization, if they lose the stain, if they lose the stain, because when they can lose the stain, whenever the bacteria have thin cell wall and outer membrane. Outer membrane is basically consists of lipids and a thin cell wall. Because of these two reasons, they can lose the primary color whenever you treat it with the decolorizing agent, that is alcohol or acetone. If that is the case, if you add counter stain, then the decolorized bacteria can can retain the counter stain. Now, if you look under the microscope, certain bacteria looks blue to purple, or certain bacteria looks uh, pink or red color. The purple bacteria, the bacteria sample which looks the purple color, they are called as a gram positive. Whereas the sample looks pink to red or red to pink, they are gram negative. Right? Hope you understand the concept. If at all you like the concept, the way we, way I have designed the concept and the diagrams and so on, try to subscribe my channel. That is Dr. PREM-PRIMER, and try to give thumbs up if you like it. If you have any comments, write to me. I'll get back to you once I see your comment. Okay, then see you again. Until then, bye. Okay, see you. Bye-bye.